This project directly led to the thesis work for one of my students who was recently awarded a Knaus Fellowship, so thank you guys for that as well. We all know that coral reefs are experiencing this period of decline, and the questions of what drives decline and what can lead to restoration or lead to recovery of systems are where the focus of most coral reef ecosystem studies are going right now. Here in South Florida, what is commonly blamed for a lot of our woes in terms of coral ecology and coral decline is water quality issues, particularly along the southeast Florida coast, along the 9 inlets from roughly St. Lucie Inlet down to Biscayne National Park. Now, although this is a public perception, strong data behind the roles of water quality and their influence on corals in Florida is less abundant than you might expect. And most of the work that is linked water quality to coral decline has been done in other regions, not along the southeast Florida coast. So these are very highly managed watersheds as well, which means we have decisions that we can make about how this water is diverted. And Tiffany spoke about this and demonstrated quite a bit of that. I knew she was going to, so I didn't have to put it in my talk. We also are constantly being told about Lake Okeechobee and its role in this system. And right now, Lake Okeechobee is certainly playing a huge role as it collects water that was deposited by Irma, and then that water is shunted into the Caloosahatchee and St. Lucie estuaries. And during times like this, Lake Okeechobee is a major driver of water quality in these systems. But it's not the only driver, and we would be remiss to characterize it as such. We also know that we have a growing population. Our whole governance model in Florida is around increasing population growth. And so we're going to have increased water needs and wastewater carrying capacity needs as well in this area. So we really wanted to focus at the northern end of this extent because folks have been doing a little bit more work further south, and because St. Lucie Inlet is directly adjacent to the biggest influence from Lake Okeechobee during these discharges. So just outside of St. Lucie Inlet is essentially the northern terminus of the Florida Coral Reef Tract. And it's at this very strong ecosystem transition zone where south of the inlet you have roughly 25 species of corals, and north of the inlet along the Florida coast you drop down to about 5. So this really is a terminus of a number of species along our coast. But it's also an interesting area because you get this transition and mix of both tropical and temperate fishes and invertebrates, making it a very biodiverse area. A lot of that influences the commercial value of this region. We have a very strong fishery focusing on kingfish, mackerel, and some snapper species as we move further out. The area is also encompassed within a state park, the St. Lucie Inlet Preserve State Park. And so a goal of the park managers was to not only understand the influences of this discharge water on their system, but also to try to quantify some of the improvements that may come as a result of heavily invested in restoration strategies throughout the state to try to improve both the quantity and quality of the water that is entering and then exiting this estuary onto the reef. This reef is not what you would classically think of as a beautiful, strong, tall coral reef with lots of branching corals. Most of the corals in this location look like these two here, Pseudodiploria clavosa and Montastraea cavernosa, corals that tend to be plating and near the bottom rather than erect and upright. So we think that's probably related to strong wave energy, but in general, this is a reef that should be considered marginal. So it's already at this northern terminus of what we know most of these corals can handle. So we have all of these different water discharges entering the area, as well as the entire local watershed influencing the St. Lucie estuary. Discharges through all these canals are, of course, managed by this very simple model from the South Florida Water Management District. If any of you have had the opportunity to take place in the weekly calls, if you have not, I would encourage you to do so. It's a very educational experience. When decisions are made to discharge water, the predominant drivers of those are the height of the water in Lake Okeechobee, as well as projected rainfall to the Lake Okeechobee watershed. That's when decisions are made to discharge water through the C44 canal. And right now, for example, C44 canal is flowing at a rate of averaging around 8,000 cubic feet per second, which means that the entire St. Lucie estuary is freshwater right now. So we expect to see some die-offs related to oysters, seagrasses, etc. within the estuary. And there's been a lot of monitoring that's gone on within the estuary, but until we started this project, nothing was being done to monitor what happens once that water exits the inlet and flows out onto the coral reef. And here's what it can look like at different times of the year. So we typically would associate some of these pulses and discharges with the rainy season or late wet season. And we do see some of those in September, for example, but it's not isolated to the rainy season. Indeed, when we have strong rain events in January and February of 2016, we saw more extensive discharges and a greater extent of the freshwater influence out onto the reef itself. And so we've used drones to map this at least 9 different times now. And one of the things that I will say from this work has been that the data generated by the drones pales in significance to the story you can tell with the drone. So we generated reports, data, publications. None of those made the impact that the video we produced utilizing some of this video, utilizing some of this footage did. So as we're developing our science plans, I would encourage all of us to think about not just the data and the outcomes, but the storytelling aspect of it as well. So our response to this problem has basically been threefold: to conduct a series of field monitoring and sensor deployments to look at the incidence of these freshwater events on coral reefs and their impact as well. To do controlled experiments, because we have this constant conflict and confounding between wet season and hot weather here in Florida. So it's difficult in the field to disentangle the relative influence of the discharge water versus the relative influence of thermal stress or upwelling events, for example. So by doing controlled factorial design experiments, we could do that in the lab. And then lastly, we wanted to really look at molecular diagnostics so that we could get at not just telling managers when a reef was already dead, but give some measure of stress so that we could indicate when corals were in a situation that needed to be rectified. So our goals of combining these three were to share our data directly and make recommendations with agency partners in Southeast Florida, to conduct graduate and undergraduate training, and then lastly, to of course do research products, papers, as well as those outreach products like the video I mentioned earlier. So for our water quality and field monitoring surveys, the water quality, we were leaning heavily on 2 existing networks, both the DB Hydro network and the Indian River Lagoon network of Lobos. So these were assets that were already in place and managed by other people like Dennis Hanasak in the back of the room that were providing us lots of information about the water quality within the lagoon. We wanted to augment that with a sensor array once you went out of the inlet onto the reef. So we deployed Odysseys. They're much cheaper, simpler than any of the instrumentation within the lagoon. They give very precise data for a short amount of time, then they fail. To all of you that are thinking about using them. But they do work for about 2 years before they bite the bullet. And then we combined that with quarterly surveys on the reef as well as the experimental techniques that I mentioned, and we'll go into more detail in just a moment. So for those controlled ex situ experiments, we were collecting corals, fragmenting them into known sizes, tracking every individual coral genotype that we were using in our experiment to control for genotype and also to predict what corals may respond better to some of these stress conditions than others. As Josh Patterson indicated earlier, the different genotypes of coral can respond quite differently to stress, or there could be trade-offs between stress and growth patterns among corals that can be important for restoration strategies. And then we exposed them to this factorial design experiment where we had elevated temperatures to simulate stress from thermal stress, as well as discharge water that was collected from within the inlet and offshore water that was our control water. And then all possible combinations thereof. One fragment from all of the different colonies that we sampled went into each of these treatments, and then all treatments were replicated 3 times across 3 different raceways. So normally, this third part I would include in our response section, but one of our actual deliverables of this grant was the development of this pipeline. So that's why I've switched over to results here rather than just our response. So optimizing this molecular analysis pipeline was the first step for this project. And basically what we've done is to work out a system that we use repeatedly in all of our studies to take coral samples for population genetics and gene expression, Then we take that coral fragment and remove the coral tissue using a water pick, separate out the Zooxanthellae or the endosymbiotic algae from the coral, look at what type of algae are there, how much chlorophyll they have, and how many there are. And then we also take that naked skeletal fragment and we 3D scan it so that we can get a really good estimate of surface area that we can scale everything back to. So looking at population genetics, we really focused on Montastraea cavernosa. It's the most common coral along this area. It's also the most broadly distributed across the Caribbean. And what we found is that among our 3 sites, St. Lucie and down into Jupiter and Palm Beach area, that we likely have 3 historical source populations, but they're all relatively well connected to one another. Indeed, you couldn't differentiate them using AMOVAs or FST or anything like that. There's this slight indication of a migration from north to south. So in other words, eddies peeling off of the Gulf Stream that can carry larvae southward. But in short timescales, within the past 10 years, the migration rates estimated from this project indicate that most of these coral populations are self-seeding. They're not sharing much larvae across populations. So there's historical evidence of connectivity, but in the near term, they're relying on themselves for population maintenance. We also saw essentially no evidence of corals that were generated by asexual propagation. We had no clones in any of these populations. So we know something now about the population structure that's there. The question is that population structure and are those different communities being subjected to this discharge stress that we think is going on? And can we relate that directly back to either rainfall or discharge? So here you can see the relative salinity of the top graph versus rainfall in the middle graph. And you'll see that pulses of rainfall are immediately followed by drops in salinity, not just in the inlet, but out on the reef itself. So that fresh water isn't just sitting on the top, it's mixing in and dropping salinity as you move onto the reef. And that can also be driven by freshwater discharges. So this is the same time period, and what you'll see is that we're getting discharge from the C23, 24, and 10-mile creek canals. But this is an interesting time where, because of local rainfall entering the C44 basin, rather than letting it run out into the estuary, that water was actually diverted backwards into Lake Okeechobee. So it's not always the case that when we have a big rainfall event in Southeast Florida, it's going to be running right out onto the reef. We do have flexibility in how that water is managed. When we looked at gene expression, and please pay no attention essentially to the figure on the left, except to say that there were differences in the expression responses of these corals that were exposed to stress. And we really focused on cavernosa here because it's the only coral that has a really strongly annotated transcriptome that is found at St. Lucie Reef. And we essentially saw no detectable differences among any of our sites, but we did find elevated stress genes during times of discharge events. So genes related to cell metabolism, tissue repair, or osmotic stress were all elevated during these discharge events, indicating that the corals were experiencing stress. One of the other interesting things here is that we wanted to look and see if there was a response among the algal cells that were living within the coral. So we looked at both of those major species, Montastraea cavernosa and Pseudodiploria. We found a couple interesting patterns. One, they don't change over time. So we're not seeing a shift in the symbiotic relationship among the corals in terms of what kind of algae they have. One of the adaptation mechanisms that has been proposed for corals is that during times of thermal stress or during times of other kinds of stress, they could switch to some other kind of algal symbiont to compensate. We don't see that going on at St. Lucie Reef. We see them maintaining what they have. But when we expose them to thermal stress, either in the field or in the lab, what we find here is that although they don't change the type that they have, the number of algal cells that are present can be dramatically impacted, as well as the chlorophyll that is contained within those algal cells. So what you'll see here is that in our factorial experimental design, while we may have hypothesized freshwater discharge playing a big role, it was really thermal stress that played a greater role than freshwater discharge in disrupting this symbiotic relationship and causing reductions in the amount of chlorophyll, which may lead to reductions in health status of those corals. So what are our conclusions? really we had this new optimized molecular pipeline. We started to apply it beyond just this study to work in Cuba, Flower Garden Banks, and Belize as well, which came directly out of this project. We know that we have this evidence of connectivity that's historical, but they may be isolated now in terms of maintenance. We know that thermal stress plays a greater role than discharge exposure. And what that really means is that we should think about timing of discharges to our reef. That if we're experiencing a strong thermal stress event, we may want to try to hold back water as long as we feasibly can in some of those areas. And so overall, I would characterize St. Lucie as a reef that's persisting but vulnerable. There's not high levels of recruitment. There's not high levels of growth. It's just barely maintaining itself at a state that is likely to be impacted during stressful events. The outreach video, which I'll give you a link to in just a minute, has been viewed by about 9,000 folks, and that directly contributed to 3 bills. So that outreach video and then some of the language that we developed out of that was shared with Martin County and Senate President Negron's office. That gained a lot of traction within the Florida Senate and led to 3 bills, both funding to look at water quality issues by FDEP in South Florida, as well as creation of a coral conservation area for Southeast Florida, and a renewed effort to try to improve the flexibility of water flow within the middle of the state. Really what we're trying to give them here is a way that they can quantify improvements when these investments are made. Unfortunately, we're going to have a new wrench in the system because a disease has just come into this area in July of 2017. We have new funding to look at both the impacts of disease as well as water quality on the system going forward. A lot of folks to thank, especially the 3 starred graduate students at the top that all contributed immensely to this project, and lots of partners both before and after the Sea Grant funding that we've received, so thank you all very much. If you want to learn more about us or to see that video, here's how you find it. Thanks.